Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor, Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. So, today we are going to see the basics and fundamentals, we can also say the concepts of targeted drug delivery systems. So, here today we are going to see the various topics on targeted drug delivery systems. So, first we will see the introduction of targeted drug delivery systems, and after that we are going to see the need of targeted delivery systems. Then we are going to see what are the advantages compared with the conventional drug delivery systems, and then we are going to focus on the various strategies of the targetings. So, today we are going to see the few major targeting systems, which are positive targeting, active targeting and the levels of targeting. As we know that targeted drug delivery is a system of delivering the drug moiety directly into its targeted body area. Now, think about the statement. So, targeted drug delivery. Now, so far we are discussing on the novel drug delivery systems. Yes, by encapsulating, by loading of our drug molecules into different carriers, either it may be in the tablets or by modifying the surface of the various nanosystems and encapsulating the drugs into various nanosystems, we are delivering the molecules, right, and we are controlling the release and we are modifying their physicochemical properties. But that will not be effective for the treatment of the life threatening diseases. And when you use some potent drugs, the story will not same when you treat for the cancer treatment. So, now when cancer comes, we know that their growth and the functions of the cells will be different. So, the targeting of cancer cells is completely different from the normal cells. If you use anti-cancer drugs for the treatment of the cancer therapy, for the treatment of cancer, we know the anti-cancers are cytotoxics. They will definitely kill your normal cells also, especially which will attack on the bone marrow and also the hair follicles, that leads to the bone marrow depression as well as alopecia, and we also see the destruction of various normal cells. So, that is why the targeting of anti-cancer molecules you cannot make in a conventional formulation, you cannot give to the patients for the treatment, because we will see lot of side effects because of non-specific targeting that leads to the various side effects. Whereas, targeted drug delivery, as we studied the statement, we delivering the drug moieties, maybe these are chemical moieties or biologicals or therapeutics that we are directly delivering into the particular site, which are the organs, cells and the subcellular levels of the specific tissues, also known as the organelles of the cells, to overcome non-specific toxic effect of the conventional drug delivery. That is one of the major limitation of the conventional drug delivery systems. So, when you target, definitely the dose of drug will reduced. With that, you will see the reduction in the side effects. So, thereby the reducing the amount of drug required for the therapeutic efficacy. So, with this target delivery system, we can target our molecules into the specific site to overcome the non-specific toxic effect of the formulations. And these are also play major role in the site specific release of the drug from the dosage form based on the pH, based on the temperature and based on the environment. So, it will release the drug. Now, the Paul Ehrlich proposed the concept of magic bullet. Now, in the picture you can see the nanomedicines or the targeted medicines will work as a magic bullet. They will go and bind to the target zone or specific zones. It will not go and bind with the non-specific sites. So, he introduced this concept. So, the statement comes, the selectively targeting a pathogen without harming the host organism. So, now we can see in the picture, you can see this is a considered as a virus and this is a drug molecule. So, this will go and these are the magic bullets which are nothing but the targeted approach which will specifically hit the microorganisms. It's the same concept implemented for the anti-cancer therapy. So, once you target your medicine anti-cancer formulation, this will only specifically will go and act on the tumor site, which will not go into the normal cells. So, this concept is adopted into the different drug delivery strategies. So, the drug is delivered to a specific location, it is called as targeted drug delivery systems. Now, here we can see there is a lot of unmet needs in a conventional drug delivery systems. So, we can see what are that. So, scientists have overcome, scientists have created the bridge to fulfill the gap of the nanomedicines. So, here you can see the unsatisfied performance of the drugs with the conventional delivery in terms of pharmacodynamic and pharmacokinetic and pharmaceutical and the pharmacotherapeutic features. So, there is a huge demand in the targeted drug delivery. So, we will see first with the pharmaceutical, as we know that the most of the API or you can say consider anti-cancer molecules and synthetic molecules which suffers with the solubility and the permeability. So, to overcome these issues, so we need to target with the proper target delivery systems. To overcome all the pharmaceutical limitations, to improve the performance, we must be convert the conventional drug delivery systems into the targeted delivery, so that our dose will reduced. Now, in terms of pharmacodynamics, the low specificity and the low therapeutic index is also one of the limitation in the pharmacodynamics. to overcome that, we need to convert the conventional delivery systems into the targeted delivery. So, pharmacotherapeutics, like the high dose required, and yes, when you use high dose it leads to the adverse effects, and definitely the patient complaints is very low. So, yes, when you translate your medicines from the conventional drug delivery to target delivery, so this will, you can reduce the dose and you can reduce adverse reaction, you can improve the patient compliance. As we know, when you target, your pharmacokinetic profile will vary compared with the conventional systems. Like you can improve the half life and also you can optimize the volume of distribution of your drugs. So, with this, so to the one solution of all these conventional limitations, by using the target drug delivery systems, we can improve the performance of the drugs. So, now here we will see the advantages of the target drug delivery systems. As we know that which are site specific, they will release at particular site on controlled basis. Yes, with that definitely it will not reach to the non-specific sites. So, there is an improvement in the efficiency. Yes, that can be also possible with the low dose of the drug, that will also help to reduce the toxicity. When you use targeted systems, by modifying their surfaces with some active targeting or by using some ligands, and you will see there is a lot of change in their absorption, distribution, metabolism and excretion profile. So, with the targeted system, we can also modify the pharmacokinetics of the formulations. Controlled biodistribution also possible, because we are targeting to the particular site based on the time interval and based on the pH and based on their tumor microenvironment, we can also maintain the controlled biodistribution with the target delivery systems. And yes, increase specificity of the localization. So, that can be possible by only with the targeted delivery. So, we can improve the specificity. If you are looking for the particular organ, by modifying with the various antibodies and with the some functionalities, definitely your carrier will go and will retain in that particular target site compared with the conventional systems. And if you reduce the dose, definitely we can also reduce the toxicity and it will not go and bind to the non specific cells. So, that leads to the reduced toxicity. And also we can reduce the dose and dosing frequencies by doing the targeted delivery systems. And yes, if you reduce the toxicity and the dosing frequency, definitely you can improve the patient compliance. This all target delivery, especially in terms of in case of infectious diseases and for the cancer treatment, these are have the greater advantages compared with the conventional therapies. So, now this drug targeting also can be classified into different strategies. Now, the first strategy we can see here, that is the active targeting and then passive targeting. This further active targeting can be classified into first order targeting, second order, third order and fourth order targeting. So, today we are going to see this part. Yes, this targeting can also possible by the biological targeting, physical targeting and also chemical targeting. And if you want to perform at a particular region, and you can also use local targeting. And if you want to see your formulation in the systemic circulation, you can also use systemic targeting strategy. And also there are the other methods, other technologies by using inverse targeting, dual targeting, double targeting, and the combination targeting also possible. So, location based targeting also possible, and the disease based targeting. So, if you want looking for some particular disease, to target the disease, you can also modify your delivery system based on the disease. So, here we are going to see first on the what is the passive targeting. The simple definition here, the passive targeting, the targeting mechanism works on the passively. So, without adding of anything into the carriers, by using the physiology of the body and the carriers, we deliver the molecules into the particular site. So, which are called as passive targeting. So, here is a one example, tumor vasculature. So, in the tumor, we know that you can see in this picture there is a irregularity in their structure. So, you will see there is a deformation and there is a change in their structure. So, scientists have hypothesized by using the EPR effect. So, what is the EPR effect? So, here when you target the cancer cells by using the nanoparticles, so these cancer cells have the different properties compared with the normal cells. Now, you can see the healthy cells here and the tumor cells. Now, you can see there is a disturbance in the epithelial cells as well as endothelial cells, and there is a high leakage in this system, and there is a poor lymphatic reflex in the tumor tissues. Okay. So, with that, when blood flows in the cancer tissues, the chances of retention and the penetration is very high. So, it is called as enhanced permeation and the retention effect. So, that is EPR effect. These type of passive targeting can be possible in only tumor cells, whereas normal cells there is no chances of the passive targeting by EPR effect. So, based on their pathophysiology, so people have hypothesized this system, the nanocarriers, because of their nanosystems, this particle will permeate through the irregularities in the cells, and they will retain for longer time compared with the other cells. So, that is the beauty of the passive targeting, and this passive targeting can be achieved by the EPR effect, also based on the tumor environment, and also by the direct local delivery. So, another type of target system is nothing but active targeting. But here what we do, active targeting, we modify the surface of the nanosystems with the various ligands, preferably antibodies, and some of the molecules. So, that will go and specifically bind to the receptors. In the picture you can see, so active targeting of the nanomedicines. Now, you can consider this as a nano system, and this can be labeled with the various ligands. Now, this after administration, this will go and bind to the cell. So, this is a cell and this is the carrier which is loaded with the drug, and this one is the ligand. So, now this ligand have a specificity to the particular receptor. So, this is called as active targeting. So, here we will target the particular cells by modifying the surface of the carriers either by the antibodies and some of the molecules. So, here is the folic acid is one example. We can modify the nanoparticles by using the folic acid. So, these are preferably bind to the folic acid receptors which are overexpressed in the cancer cell. So, consider these are the active targeting systems, and these will specifically bind to the surface of the cells, and that will undergo the internalization, and further this particle transport into the endosomes, and there your drug will release. So, further the drug will show its action. So, that all information you can read it here. Interaction between the ligand and the target can induce the intramembrane folding leading to the nanoparticle internalization via receptor mediated endocytosis, and that leads to the transfer into the nuclear endosome, further the drug release. So, this is the active targeting mechanism. You can take polymers, polymeric nanoparticle, you can take lipid nanoparticle, you can also take any type of inorganic nanoparticle, you can modify the surface by using the ligands. So, here you can see the lectin carbohydrate interaction, that is a one technique, ligand receptor interaction, antibody antigen interaction. So, these are the few examples for the active targeting. And here also you can see there are other targeting approaches. So, here is the stealth, that means you need to modify the surface of your particle by using the hydrophilic polymers. So, it will avoid the phagocytosis mechanism, opsonization mechanism of the your nanoparticles. So, whenever you use hydrophobic particles, so the opsonin will come and it will undergo the opsonization, and it further undergo the RES system, reticuloendothelial system. This can be avoided by using the hydrophilic polymers, preferably polyethylene glycol, that will reduce the RES uptake of the nanoparticle system. With this, you can enhance the T half of your molecules or nanoparticles in the blood. Now, this we already seen receptor mediated, this is a folate receptor, we can target the folate molecule, transferrin receptor, we can target the transferrin receptor by using transferrin, asialoglycoprotein receptor, this can also be performed by using various carbohydrates and also by the adhesion. Now, antibody mediated, we have seen in previous slide that HER receptor can be targeted by using the antibodies, and stimuli response also possible by using hyperthermia, magnetic properties, ultrasound properties, and also the pH and temperature and the temperature. So, pH, we know that cancer cells are the pH is near 5.5. So, to target this, what you need to do? You need to select pH sensitive polymers can be used to target the cancer cells. If your system is temperature based or hyperthermia, so here what you can do? You can use temperature sensitive polymers. So, at that particular temperature your polymer will release the drug. similar to the pH sensitive also. Now, here we are going to see the levels of targeting. Now, we will see these are the classified based on their targeting mechanism. So, here we will see first one is the first order or organ based targeting. So, we can target your nanosystems or your formulation by targeting the organs. So, if you perform or if you target the your systems based on the organ, so it is considered as a organ based or first order targeting. So, we can also target the liver, we can also target the brain and other organs, even the pulmonary also possible. So, these are called as a the first order or organ based targeting. Secondary order targeting is that is cellular targeting. We will target particular cells. So, for example, tumor cells. So, to avoid the toxicity on the normal cells, we use various types of the strategies to reach the tumor cells. So, that is called as secondary target. Similarly, it can also possible for the hepatocytes. And the third one is the tertiary order organelle based targeting. Here these are the highly approachable targeting, and yes, here you can also target the nucleus and the mitochondria and other organelles of the cells. So, all these cases the targeting approach will vary. And the fourth one is you can further go into the deeper cells, that is the macromolecular targeting, which is called as a fourth order, that is we can also target the DNA and the proteins by using this targeting strategy. So, organ based targeting, cellular targeting and then organelle based targeting, and third one is the last one is the molecular targeting. So, with all these systems we can also target the various types of the diseases. So far we have discussed on what are the targeted drug delivery systems, and also we have seen the advantages of the targeting drug delivery system. Then we have seen the various strategies of the drug delivery systems, and in the last we have discussed the various strategies of the targeted drug delivery systems. In that we have discussed the passive targeting strategy, active targeting strategy, and also we have discussed level of targeting. In that we have seen 4 different levels of targeting. So today we are going to stop the lecture here, and in upcoming classes we are going to discuss on the details of the various targeting systems. Thank you so much.